In this video, we're going to be talking all about DNA, what it looks like and what it does. Before we get started, I wanted to mention that these are the scientists, Watson and Crick, that discovered the actual structure of DNA, what it looks like, what it's made up of. So this is the model of DNA that they created while they were doing this research. And I got to actually see pieces of this original model when I was visiting London. The Science Museum had an exhibit on it, and I was super excited. So I just wanted to include that because that was a couple summers ago that I got to see it, and it was super exciting. So when we're talking about DNA, it looks like a twisted ladder or a spiral staircase. We call it a double helix, and it's made up of two complementary strands, which means that they are not identical to one another, but they do pair up. So if you notice in my picture, the purple and the green always go together. Every time I have purple, I have a green next to it, purple and green, and then the blue and pink are kind of the other matches. So they have a pair that they always match up with. So they complement one another by going with their pairs. The backbone, which is like the yellow part of my picture here, is made up of sugars and phosphates. Okay, in this picture, I call it like a condensed DNA diagram because the backbone is condensed into one piece, even though it's actually made up of sugars and phosphates. So the actual full picture would look a little bit different. And the rungs of the ladder, so like the actual pieces of the ladder that you would climb up, those are called nitrogen bases, and there's 4 different kinds of nitrogen bases that you can have. So you can have adenine, guanine, cytosine, or thymine. Oftentimes we'll abbreviate them just to the first letter of the word. And like I was mentioning before, there are pairs. So adenine and thymine always pair up, and guanine and cytosine always pair up, and you guys can see the pairs here in their actual molecular structure. They are organic compounds, so they have a similar structure to when we were doing organic chemistry. If you're having a hard time remembering which letters pair up, the ones that are made up of straight lines, so A's and T's have really straight lines in the letter, and G's and C's are kind of more curvy. So the curvy lines go together and the straight lines go together. We also have other words that we can use to describe the nitrogen bases. So adenine and guanine, they are both made up of 2 carbon rings. So you see we have our G and our A have 2 carbon rings and 2 carbon rings, so we call them purines, and they're the larger of the 2 types of nitrogen bases. We also have pyridamines, and pyridamines are made up of just 1 carbon ring, so they're the smaller of the 2. I use the trick, purines is a shorter name, so it's the larger molecule, and pyridamine is a longer name, so it's the smaller molecule. It's like the opposite of what you would think. So also DNA, if we talk about 1 nitrogen base, 1 phosphate, and 1 sugar, that's a subunit called a nucleotide, and it wants you to draw a picture. So our phosphate is a circle, our deoxyribose sugar is a pentagon shape, and then we have a nitrogen base. Remember, there's 4 different options, and they would all be shaped differently. So this just happens to be a triangular shape. There would be one that was like the opposite of this that would pair up with it, and we could have other shapes as well. So to make up a strand of DNA, you need a bunch of nucleotides bonded together. And in this next couple of slides, I'm going through the review questions. So what I would like you to do is I would like you to pause each time and try to do the question on your own, and then press play when you are ready to see the answers. So you can pause right now for this one, you're labeling. So it asks you to label deoxyribose sugar and a phosphate. And then you have to label the adenine and the guanine and the cytosine and the thymine. So it doesn't really matter which ones you picked as long as you paired them up correctly. So A always goes with T. And T is a pyridamine, so it's the smaller one. So you should have T labeled as the smaller of the 2. If you picked this one as the A and T, that's fine. And then C and G as well have to pair up. G is the bigger one, the purine. So it would be the bigger of the 2 options, and C is the smaller one. It's a pyridamine. So that is how we would label that. Again, you could have these flip-flopped, and that would be fine as long as you have the smaller one and the bigger one in the right spot. Questions 2 and 3 are asking you to pair up the letters of DNA. So if we take this combination of DNA, you have to pair up the A's with the T's and the G's with the C's in the complementary strand. So it wants you to rewrite the DNA on the other side of DNA. So if it says A, it would say T, T, T. So now you can pause and finish the rest of the strand. And this is what it should look like. So T, 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 this should go with A, G, C, G, T, A, G, A. Second question, same thing. You guys can pause and write out the complementary strand. It should look like this: C, 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 T, T, A, G, G, T. The last three questions in the review questions are going through and talking about the nitrogen bases. So in question 4, it says that we have 24% adenine, which means that if we're using percentage, we're talking about out of 100, we have 24 letters that are A. A's match up with T's, so I know that I have to also have 24 T's. So if I have that, 24 plus 24 is 48. I've used up 48 of my letters in my strand of 100, so that means I have 52 letters left over. But I still have 2 letters to account for. I need to figure out how many guanine there are, but I also need, in turn, to figure out how many cytosine there are. So 52 divided by 2, because I still have 2 letters to figure out, is 26. That means that I have 26 cytosines, and finally 26 guanines, which is what the question is asking for. So if you went back and added 24 plus 24 plus 26 plus 26, it should equal 100. Question number 5, you guys can do this one on your own, so I'll just get you to pause and you can try it out yourself. So the answer here should have been that we had 34 cytosines, which means we have 34 guanines. That's part of the questions, what percentage of the molecule is guanine. But I also have to figure out thymine. So I'm going to add them together. That means I've used up 68 of my letters, which means I have only 32 left. And that divided by 2 is 16. So 16 thymines and 16 adenosines. It only asks about T's and G's, but you kind of have to figure out all of them anyway. So I thought I would just lay them all out anyway. The last question, again, try it on your own. We have 28% guanines, and it's asking for pyridamines. So pyridamines, remember, are T and C, the smaller of the 2 types of nitrogen bases. So we need to figure out basically all the letters in order to figure out pyridamines. So first it tells us I have 28 G's. So that means I have to have 28 C's as well, which means I've used up 56 of my letters, which means I have 44 left over, which means that I have 22 A's and 22 T's. So my pyridamines are C and T, so that would be 28 plus 22, and that's 50 in total, which makes sense because half of my strand should be made up of the larger molecules and half of them should be made up of the smaller ones. So it's also asking about functions of DNA that we need to know about. So DNA, I'm sure you know, is made up of genes, and we talked about that a couple weeks ago, and we use it for passing on traits, but it also really importantly is used to make proteins. So proteins are really, really important things in our body, like things that control what we're doing. All of the levels of stuff that's happening in our body are controlled by proteins. And our DNA is like the recipe book for the proteins. How do we make them? Our DNA tells us. So proteins are things like catalysts, like enzymes, okay, things that carry nutrients, things that create muscles and stuff like that. And about 30 to 40% of your body is made up of proteins, but everything else in your body is actually like responds to proteins. So if it's not made up of protein, it like interacts with protein. So basically, protein is super, super important for your body to function properly. So that specific combination of A's and T's and G's and C's in your DNA is basically like sentences explaining to your cell how to make the certain proteins that they need to make, which is super important. Our DNA also serves as hereditary material. So that means that slight changes in the proteins that your body is creating makes you look a little bit different. So example, blue eyes versus brown eyes, it's just a little bit of different nitrogen bases, so maybe an A instead of a T out of thousands of nitrogen bases that are on the eye color gene. The reason why our DNA is all different is due to mutations. So we're going to talk in a couple days about DNA replication. So sometimes there might cause mutations when the DNA is replicating. Some of the letters might not get transferred over properly. There's something called crossing over when we create an egg or a sperm cell. Your DNA actually exchanges between your mom and your dad's DNA that you have in your cells will kind of exchange. So you have just so many different options of things that could be carried in your egg or your sperm. And then also just the fact that one sperm and one egg come together and they're all carrying different genetic informations. There's so many different possibilities of sperms and eggs that could go together, creating different combinations of genes due to sexual reproduction. So that is basically all we have to know about DNA for today. Hopefully it makes sense. If not, please feel free to ask me any questions, and I'll talk to you guys later. Bye.